Okay, hello everyone. So welcome to our first cell bio video. Today we're going to be talking about gene regulation in prokaryotes specifically. And so we'll get right into it if I can control my computer. Okay, so right in front of you, this visual is called an operon. The reason I'm showing you it is because an operon is one of the simplest regulatory systems found in an organism. Specifically, this organism would be bacteria and some viruses. So a typical operon consists of a group of these structural genes that code for enzymes or proteins involved in a metabolic pathway. These genes are located under the control of one promoter and one operator. And so you'll see in future slides how the promoter and operator works, and I actually have definitions for them in the next slide, but they pretty much have regulatory power over these cluster of genes that are going to be transcribed into the single mRNA transcript. And so this is the overall view. I'm going to switch back to it in a little, but I have to give you definitions first. So terms to know. The structural genes that we saw, A, B, C, D, those code for the protein of interest, or if they're going to be enzymes. Next, we have the operator site. And so this operator site is going to be a little upstream of the structural genes. And so it's a non-transcribable region of DNA that is capable of binding a repressor protein. You're probably wondering what a repressor protein is, and we'll get into that later in this video. But the operator site is right here, ready for a repressor or activator protein to bind. The promoter site, it provides RNA polymerase with a place to bind. So this is similar to what we find in eukaryotes. It's a little upstream to the operator, but RNA polymerase will bind here and see that the start of transcription is here and do its duty. The last thing, way upstream, is going to be your regulatory gene that codes for the protein known as a repressor that we just talked about and we'll talk about later. But that's pretty much a coverage of the simplest parts in an operon. We'll get into the specifics of how it functions. So there are two types of operons. There's an inducible system and a repressible system. The inducible system is when transcription is normally turned off, but it can be made to turn on given a particular signal. So a repressible system is normally on, which means that transcription is continuous, but can be made to turn off given a particular signal. So let's get into these specifics. I'll be repeating those two definitions throughout the video so that it's ingrained in your mind by the end of this. But those are the foundational concepts that you need to know to apply anything about this concept on any question, on any standardized exam, etc. So the first example I have here is the lac operon. The lac operon is an example of an inducible system. So what does that mean? That means transcription is usually turned off, but it can be made to turn on. So look into that word, inducible. If something is inducible, then you're allowed to turn it on, but its normal state is that it's off. So in this figure, we see that a repressor, something that we talked about before, something that the regulatory gene encodes, produces, and that repressor can go and bind to the operator here. But when the repressor binds to the operator, that blocks RNA polymerase from transcribing the genes. So imagine the repressor as a roadblock in the way of transcription of the lac genes. And so in its normal state, the lac operon is an inducible system. Therefore, it's normally turned off. The repressor is blocking RNA polymerase from binding, but it has the opportunity to be turned on. So let's get into that opportunity. Here, we see that lactose is able to bind to the repressor and move it away from the operator site, disallow it to bind to the operator. In that way, RNA polymerase is allowed to go ahead and transcribe the lac genes at a slow rate. But what you need to take away from this is that transcription is normally turned off. In this case, the repressor is bound to the operator, disallowing RNA polymerase to transcribe the genes. but it can be made to turn on. An inducer binds to a repressor, moves it away from the operator, and allows RNA polymerase to transcribe the genes. And so that is a foundational concept you need to know about inducible systems. And a very common example would be the lac operon. So let's get into a repressible operon. So in this case, we're using the TRP operon, or I like to call it TRP operon. So the TRP operon is a repressible system, meaning that transcription is usually on. In its normal state, transcription is usually on. RNA polymerase is transcribing continuously, but it can be made to turn off. So where do you see these types of systems? In places that negative feedback is needed. And so in this repressible system, which a common example would be the TRP operon again, transcription is usually on the go. RNA polymerase has the freedom to transcribe and transcribe. The repressor isn't really vibing with the operator site. It's just they don't connect in repressible systems. But something can happen. The product of these genes can go ahead and bind to the repressor, making it a co-repressor. And so in this case, tryptophan would be the co-repressor. It can bind to the repressor, and that would allow the repressor to suddenly have an affinity for the operator site. And so when it binds to the operator site, it disallows RNA polymerase from going ahead and transcribing. So what do we see again? I'm going to reiterate the description of repressible systems. Transcription is normally on, but has been turned off or can be turned off when a co-repressor binds to a repressor, which in turn binds to the operator site, blocking transcription. So now we know the foundational concepts behind what an inducible operon is and a repressible operon is. Now, in all these examples, we've talked about a repressor being the other molecule present and regulating the system. That's not always the case. So we're going to switch to the whiteboard and see what happens when there's a repressor. Again, recover that, and then see what happens when we add an activator. Let me see if I'm recording. So we've switched to the whiteboard. I want to give you a quick overview of inducible versus repressible, but I'd like to clarify one thing. I added negative in front of both the system names. The reason I say that these are both negative inducible and negative repressible is because a repressor is active. Let's say an activator was in its place, which we'll talk about further in the video. In that case, the systems would have positive in front of it. And so the negative versus positive that's in front of the system name refers to whether a repressor or activator is present. And then inducible versus repressible we'll continue to talk about. So in an inducible system, we've talked about it already. It's normally turned off, but can be made to turn on. It's inducible. So since it's normally off, that means that the repressor has to be active. It has to bind to the operator site, thereby inhibiting RNA polymerase from transcribing these structural genes. Since it can be made to turn on, we see that a substrate can come in, bind to the repressor, and inactivate it, allowing RNA polymerase to come in and transcribe the structural genes, therefore turning transcription on. And so again, we know the foundational concept of an inducible, where transcription is normally turned off in the system but can be made to turn on. Now let's go ahead and go to the repressible system. Transcription is normally on. There's continuous transcription with these systems, but it can be made to turn off in certain situations. And that's what you see in negative feedback situations. And so in a repressible system, you see that the repressor, since it's a negative repressible system, the repressor is just going to be inactive. It won't bind to the operator site. It has no affinity for the operator site. And our RNA polymerase is having a dandy time transcribing. Whereas if you want to turn it off, if you want to repress the system, it's repressible. If you want to do that, you can take a product that's made in the whole system and it can act as a co-repressor, bind to the repressor, activate it, and then the repressor and co-repressor complex will be able to bind to the operator site, disallowing RNA polymerase from transcribing. And so transcription is turned off. And so really it's as simple as that. You just need to keep clear what repressible means and what inducible means and see where to go from there based on what other molecules are present in the environment, such as a repressor or an activator. But that's pretty much all I have for the whiteboard. We're gonna go ahead and switch back to the PowerPoint so I can explain what happens in positive inducible and positive repressible systems. Okay, so now we're gonna go deeper and we're gonna say, well, what if it's a positive inducible system? What if it's a positive repressible system? Positive meaning that an activator molecule is present. Don't get too caught up in the detail. We already know the foundational concept. Like I said, it just takes some application knowing that concept. So we know that an inducible system is normally turned off, but can be made to turn on. If an activator is present, it's going to be inactive when the inducible system is in its normal state. However, it can be made to turn on, remember? So if an inducible system wants to turn on, a substrate will bind to the activator, make it active, and it'll bind to the operator site and induce transcription. That's a quick coverage of a positive inducible system. In positive repressible, what do we know about repressible? Repressible is that the transcription is normally turned on, so the activator is going to be active in binding to the operator site and promoting that transcription, but the repressible system can be made to turn off. The product will come over here, make the activator inactive when the repressible system wants to turn off. And therefore, if the activator is not binded to the operator site, then RNA polymerase will fail to bind and transcribe the genes. So what do we need to know? We need to know these two definitions to apply anything further about operons. Once you get those concepts down, come back to this figure and explain it to yourself in detail. You should not have to memorize it. You should be able to pinpoint what a positive versus negative system does if you know the foundational concept of the system itself. So hopefully this video helped. I know that you guys are past gene regulation from exam 1, but this helps in so many future classes such as genetics, any other biology class, etc., or any standardized exam. Drop in the comments below if you want to see any other topics. I'm just kidding. Just email me if you want to see videos on other topics, and we'll see if I can push the content out. But yeah, thank you for watching.